is just get rid of the tumor, okay? Reduce the mass, get it off as much as possible. I mean, try it. So surgery. If you can't, then try to kill the tumor cell, okay? And the killing could be brought about by indiscriminate bombardment of the drug, for example, or that's called as a carpet bombing, okay? So in a carpet bombing situation, you kill everybody. So it's a bombardment of the total area. Excuse me, please, Razak, pay attention, and your fellow. Okay, so carpet bombing is actually indiscriminate, leads to killing of the tumor cells, of course, but
also normal cells.
Also destruction of the normal cells. Okay, so killing of the tumor brings about remission or cure, and killing of the normal tissue brings about adverse effects. Okay, so adverse effects are resulted. Okay, so there are three main ways of doing this: chemotherapy, radiotherapy. We will be interested in chemotherapy for this lecture and the next one. Now you can also devise some drugs which can do target killing. So rather than carpet bombing, you can kill. You can target and kill. Okay. So for example, immunotherapy or immunoglobulins for certain types of cancers. Okay. Much debate or much research has been focusing on the radical or radical cure. So cure from the very roots of the genesis of cancer. So an emphasis on correction of the wrong gene. Okay, so gene therapy. But so far it's not that successful. Okay, so let's focus on the classification of the chemotherapeutic drugs. Okay, so forget about radiotherapy, surgery, we are not interested for the time being. We will focus chemotherapeutic drugs, anti-cancer drugs. Cytotoxic drugs. Okay, for example, alkylating agents and related compounds, antimetabolites, and natural products. Now, natural products is actually is a group of heterogeneous compounds. Okay, heterogeneous in terms of their chemistry, heterogeneous in terms of their pharmacokinetics, heterogeneous in terms of their adverse effect profile, heterogeneous in terms of their mode of action, but common in terms of their origin. Okay, the sources from where they are derived from, they are from the natural resources. For example, plants, bacteria, etc. Okay, so they share the common property of being natural in origin rather than synthetic. Okay, so alkylating agents are the ones which are the most devastating. Then antimetabolites, they focus some metabolic pathways in the cell. We'll see how. Okay, the second group of drugs are hormones. Okay, so if a tumor is responsive to hormone or hormone dependent, then you can introduce an inhibitor of that receptor of the hormone to bring about remission, for example. Or if the hormone acts against it, for example, prednisone. You know that immune cells, okay, they get suppressed with high doses of corticosteroids. So corticosteroids are immunosuppressants, right? Yeah, they are, but focus, what I'm discussing with you. So the corticosteroids or glucocorticoids, they are immunosuppressants, and one way of doing this is suppress the production of B cells and T cells, okay? That means in terms of cancer of the lymphocytes or blood cells, corticosteroids could be used, okay? So now this is an agonist rather than an antagonist. So you are using prednisone to induce remission in certain leukemias and lymphomas. Okay. So immunoglobulins, we already discussed that they are gamma globulins generated against certain types of cells and certain types of malignancies. We will see how.
Doctor?
Yes, please.
Sorry, what were the examples for hormones?
I already gave you one example, corticosteroid. There will be a number of others we will see in the coming slides. Okay, so alkylating agents and related compounds are the ones which are the most devastating, and they originated from the nerve gases. Okay, the gases which Europe and USA they produced against humans. Okay, to kill each other wherever possible, and during the world wars, and they found that soldiers who were subjected to those war zones or those agents, if they had a leukemia, their leukemia, for example, responded well to those, sort of gases. So there was a theory, maybe these drugs are effective against cancer, some sort of cancers. So from that, the whole plethora of alkylating agents they grew up. Okay, and currently we have at least 5 groups. Okay. And the 6th group is a little different, but works the same way. So one is nitrogen mustards. The most famous group is cyclophosphamide and its sister ifosfamide. Cyclophosphamide is part of your drug list, so we will focus that only. Ethylenimines and methylmelamine, these are the ones which is exemplified by thiotepa, not part of your drug list, so you can leave that. Alkyl sulfonates, busulfan, part of your drug list. Nitrosuria, so the blue ones are the ones which are part of your drug list, okay, available on Moodle. So nitrosurias are the mustines, carmustine, lumustine, simustine, okay. And then streptozocin, so lumustine is representative of this group in your drug list. Triazines are not at all represented in your drug list, so you can leave them out. Yes, please.
In the last exam, we had drugs listed first generation and second generation, and we were asked to answer the clinical uses of these drugs, and nothing was mentioned about it.
At the moment, we are focusing on a different topic. So let's focus first. So any such question we can deal at the end. No problem.
I mean, for this
I already said, the drugs marked blue are those included in prescribed drug list. So you have to focus the blue ones, okay? So anti-metabolites, purine analogs, okay? 6-but-captive purine and azathioprine. Yes, please. Any, chirruping, just like sparrows and birds, doesn't suit you. You are humans, so humans shouldn't chirrup, okay? So purine analogs are the chemical compounds which resemble some purines, okay? You know what are purines, I hope.
Yes.
Purine, pyrimidine, yes. So those are
DNA.
the nitrogenous bases which take part in synthesis of nucleotides, which ultimately form DNA and RNA. Okay? So purine analogs. The only example in your list is azathioprine as an immunosuppressant. We will deal in detail in the 3rd lecture. Okay? Not in these lectures, but I would say pay some attention to 6-but-captive purine. That is important in terms of certain hematogenous cancers. Okay? Then pyrimidine analogs are more important. In fact, 5-FU and ARA-C. ARA-C is 5-FU is 5-fluorouracil. Okay? 5-FU. Okay? You can also call it fluorouracil. Okay? So in some books, it's 5-FU. In some, it's written fluorouracil. Okay? So they don't bother to write 5. Okay? So then ARA-C or cytosine arabinoside or cytarabine. There are 3 names for the same drug. So don't be confused if you follow one book. If it is aracy, okay. If it is cytarabine, okay. If it is cytosine, arabinoside, still okay. It's the same. They're talking about the same thing.
Stop chirping.
I really appreciate your female colleagues. There was a pin drop silence, and they were very well behaved in terms of attending the lecture. So it was a marvelous, I would say, marvelous experience in terms of females, pin drop silence, and gentlemen, no silence. Okay. So, this is in contrast what is being preached upon, Yeah. So you have already proven it wrong. So folate, bravo. So folate analogs include methotrexate, the famous drug, okay, methotrexate, and part of your drug list, okay. Now, we would discuss the heterogeneous group of natural products, okay, as an analog. So, the most important group is antimitotic drugs, okay, the four most important in this group. Antimitotic drugs, the periwinkle alkaloids or vinca alkaloids, okay, periwinkle is have you ever seen the 5-petal pink small flower or white 5-petal flower? Periwinkle. I mean, it grows very often wherever abundant water is available.
We live in a desert.
Okay, maybe not in this part of the world. So periwinkle is a very commonly grown flower. Okay, so vinca alkaloids actually belong to that. Paclitaxel, the famous brand Taxol, in terms of the breast cancer. So paclitaxel is also another compound in this group. So vinca alkaloids and paclitaxel, they differ in their little difference in their mode of action, but both of them target the mitotic spindle. Okay, so we'll see in the next lecture how. Topoisomerase, you know what are topoisomerases, right?
Topoisomerases, yeah. DNA.
So they help in prevention of highly twisted supercoils. Okay, whenever the replication starts, the leading strand and the lagging strand, if you remember. Yeah. So as the replication bubble spreads on both sides, there could be supercoil, okay? Highly twisted DNA. So to unwind that, we have certain group of enzymes, okay? And those are called topoisomerases. 1 and 2. So topoisomerase 1 inhibitor, okay? These are camptothecin is another plant. So there are certain alkaloid from these, and luckily they are part of your drug list. So topoisomerase 2 inhibitors, especially toposide, okay, so these are another plant alkaloids, epipodophyllotoxins, and they could be used as topoisomerase 2 inhibitors. The fourth group are antibiotics. What are antibiotics, by the way?
Produced by organisms. Microbes to kill.
Sure. So classical definition is a substance produced by a living organism against another living organism. Okay. So these are antibiotics, and you can identify them by mycins, and some other suffixes. Okay. So actinomycin D, also called deactinomycin, also called dactinomycin. Okay, so actinomycin D, you can bring in the first place, so deactinomycin. Okay, or forget about the hyphen and it becomes dactinomycin. Okay, so one and the same thing. Then anthracyclines are a group of